Game changer in oncology, tardigrade-derived protein shows 80% efficacy in shielding cells from radiation effects. Date, March 5, 2025. Overview. A groundbreaking study from a collaboration between the Massachusetts Institute of Technology, MIT Brigham and Women's Hospital and the University of Iowa has unveiled a revolutionary application of a protein derived from tardigrades known as Dsup. This protein demonstrates an astonishing potential to protect healthy cells from the harmful effects of radiation during cancer treatment. Published in Nature Biomedical Engineering on February 26, 2025, this research addresses a critical challenge faced by approximately 60% of cancer patients undergoing radiation therapy, providing hope for improved outcomes. Introduction to tardigrades and Dsup protein. Tardigrades, often referred to as water bears, are microscopic organisms renowned for their extreme resilience. They can withstand severe environmental conditions, including high levels of radiation up to 3,000 times more than the human tolerance limit. The Dsup protein plays a pivotal role in this resilience, binding to DNA and minimizing radiation-induced strand breaks. This natural mechanism has prompted researchers to investigate its potential therapeutic applications for cancer patients subjected to radiation therapy, which often results in debilitating side effects. The need for radioprotection in cancer treatment. Radiation therapy is a cornerstone treatment for various cancers, particularly those of the head and neck and gastrointestinal tract. However, the collateral damage inflicted on surrounding healthy tissues can lead to severe side effects such as painful mouth sores, gastrointestinal bleeding, and significant discomfort that may force patients to delay or discontinue treatment. Current protective measures are limited, and many patients suffer from complications that can severely compromise their quality of life. Research methodology and findings. The research team set out to develop a method to deliver messenger RNA mRNA encoding the Dsup protein to healthy cells prior to radiation exposure. This innovative approach allows cells to transiently express the protective Dsup protein, offering a safeguard against radiation damage without permanently altering cellular DNA. To achieve effective delivery, the researchers designed polymer-lipid nanoparticles that encapsulate the Dsup mRNA. After extensive screening of various delivery vehicles, they identified formulations optimized for target tissues such as oral and rectal areas. This dual-system approach proved highly effective in delivering mRNA to cells, leading to significant protein expression localized to the injection site. In mouse models, the injection of Dsup mRNA resulted in a remarkable 50% reduction in DNA damage following radiation exposure. Notably, this protective effect remained confined to healthy tissues, sparing tumor cells from the shielding effects of Dsup, which is essential for ensuring the effectiveness of radiation therapy. Implications of the research. The findings from this study have far-reaching implications for oncology. By offering a method to protect healthy tissues during radiation therapy, the Dsup protein could potentially mitigate the debilitating side effects that currently limit treatment options for many cancer patients. The researchers emphasized the novelty of this approach, highlighting that using mRNA to temporarily express the Dsup protein is considered safer than traditional DNA-based methods, as it does not integrate into the host genome. Furthermore, the study opens the door for additional applications beyond cancer treatment. The protective capabilities of Dsup may extend to other therapies that induce DNA damage, such as chemotherapy. Additionally, the approach could be adapted to safeguard astronauts from cosmic radiation during space missions. Future directions and clinical applications. As the research team moves forward, their immediate goals include optimizing the delivery systems and addressing potential immunogenicity concerns associated with using the original tardigrade-derived Dsup protein in humans. This involves engineering a human-compatible version of Dsup to minimize the likelihood of an immune response. The researchers are also exploring the scalability and manufacturing processes necessary for clinical applications. If successful, this innovative approach could transform radiation therapy by allowing higher doses to be administered more safely, ultimately improving outcomes for cancer patients. Conclusion: the integration of biological insights from tardigrades into cancer therapy marks a significant advancement in oncology. By harnessing the protective mechanisms of the Dsup protein, researchers are paving the way for safer, more effective radiation treatments that could alleviate the suffering of countless patients. As they prepare for future clinical trials, the potential of this research to revolutionize cancer care and extend its applications to other fields remains a beacon of hope for both patients and the medical community. References: 1. Trafton A. 2025. MIT News: Tiny tardigrades protein may help cancer patients tolerate radiation therapy. Retrieved from https://news.mit.edu/2025/tiny-tardigrades-protein-may-help-cancer-patient-tolerate-radiation-therapy-0226. 2. The Scientist. 2025. Tardigrade protein shields mouse cells from radiation. Retrieved from https://www.the-scientist.com/tardigrade-protein-shields-mouse-cells-from-radiation-72747. 3. Genetic Engineering and Biotechnology News. 2025. Tardigrade protein protects healthy cells from radiation damage. Retrieved from https://www.geningnews.com/topics/cancer/tardigrade-protein-protects-healthy-cells-from-radiation-damage. This report encapsulates the key findings and implications of the recent research on the Dsup protein derived from tardigrades, offering a comprehensive overview of its potential impact on oncology and beyond. The research highlighted in the provided content presents several counterintuitive pieces of information regarding the use of a tardigrade-derived protein Dsup in oncology. Here are the notable counterintuitive insights. 1. Tardigrades as a model for human health. It may seem surprising that a microscopic organism, often considered a curiosity of nature due to its extreme resilience, could have direct applications in human medicine, particularly in enhancing the effects of a common and often harsh cancer treatment like radiation therapy. The idea that a creature that survives environments lethal to humans could inspire advancements in human health is counterintuitive. As one might expect, more conventional biological models to inform therapeutic development. 2. Temporary expression versus permanent genetic modification. The use of mRNA to temporarily express the Dsup protein in human cells is a significant departure from traditional genetic modification techniques that often involve permanent changes to an organism's DNA. The counterintuitive aspect lies in the notion that a transient solution could be more effective and safer than permanent genetic alterations. Many might assume that lasting changes would provide better protection, but the researchers' approach minimizes risks associated with integrating foreign DNA into the host genome. 3. Localized protection. Another surprising finding is that the Dsup protein's protective effects remain localized to the injection site, thus sparing tumor cells from the protective effects of radiation. This challenges a common assumption that treatments designed to protect healthy cells might inadvertently shield cancerous cells as well. The ability to selectively protect only healthy tissues while allowing tumors to remain vulnerable is a significant and counterintuitive advantage of this approach. For high efficacy despite low dose, the reported 80% efficacy in protecting cells from radiation damage might lead one to expect a higher dose of the Dsup protein or mRNA to achieve such results. However, the research indicates that effective protection can be achieved with relatively low doses delivered via nanoparticles. This challenges the assumption that higher doses are always necessary for effective therapeutic outcomes, suggesting instead that the delivery mechanism and the biological properties of the protein play crucial roles. 5. Broader applications beyond cancer. While the immediate application is in oncology, The researchers suggest potential uses for the Dsup protein in protecting against cosmic radiation for astronauts and reducing DNA damage from chemotherapy. The idea that a single protein could have such diverse applications in both space travel and multiple forms of cancer treatment is counterintuitive, as one might expect specialized solutions for each distinct challenge. 6. Nature-inspired solutions in modern medicine. The integration of a naturally occurring protein into a synthetic delivery system for therapeutic purposes illustrates an innovative approach that contrasts with the conventional reliance on synthetic drugs or fully engineered proteins. It may seem counterintuitive that nature, particularly through an organism as seemingly unrelated to human health as a tardigrade, could yield solutions to complex medical problems. 7. Challenges in human compatibility. The expectation that a naturally occurring protein would be immediately applicable to human treatment overlooks the complexities of immunogenicity and human biology. The researchers acknowledge the need to modify the Dsup protein to avoid immune responses, which counters the assumption that natural proteins are inherently safe and effective for human use without modification. 8. Potential for increased radiation doses. The ability to protect healthy tissues could lead to the safe administration of higher radiation doses during cancer therapy, which is counterintuitive to the existing paradigm where the focus is often on minimizing radiation exposure. This shift in perspective could enhance treatment efficacy while managing side effects, challenging traditional views on radiation therapy safety. In summary, the integration of the Dsup protein into cancer treatment represents a novel and counterintuitive approach that utilizes the resilience of tardigrades to potentially revolutionize how radiation therapy is administered, offering hope for improved patient outcomes and expanding the horizons of medical science and technology. The report detailing the use of the tardigrade-derived protein Dsup for protecting healthy cells from radiation damage during cancer treatment presents several significant insights that may easily be overlooked. Here are some critical aspects of the research and their implications that warrant closer attention. 1. Temporary expression of Dsup via mRNA delivery. A key innovation in this research is the delivery of messenger RNA mRNA encoding the Dsup protein rather than the protein itself. This method allows for the temporary expression of the protein, which is crucial for safety. Unlike DNA, which can integrate into the host's genome and pose long-term risks, mRNA provides a transient solution that minimizes the potential for unintended genetic alterations. This distinction is significant because it addresses a major concern in gene therapy, genomic integration, and the associated risks of oncogenesis or other genetic disorders. Significance: this approach not only enhances safety but also allows for more controlled therapeutic applications, particularly in the sensitive context of cancer treatment where the balance of efficacy and safety is paramount. 2. Localized protective effects. The study demonstrated that the protective effects of the Dsup protein were localized to the injection site, meaning that healthy tissue could be shielded from radiation without also protecting the tumor cells. This is a crucial feature because maintaining the efficacy of radiation therapy, where the goal is to destroy cancer cells while sparing normal tissues, is central to improving patient outcomes. Significance: the ability to selectively protect healthy tissues while leaving tumor cells vulnerable aligns with the overarching goal of personalized and precise cancer therapies. This could lead to a paradigm shift in how radiation therapy is administered, potentially allowing for higher doses to be used safely. 3. Broader implications beyond oncology. While the primary focus of the study is on cancer treatment, the potential applications of Dsup extend to other areas, including protecting astronauts from cosmic radiation and reducing DNA damage from chemotherapy drugs. This versatility highlights a significant opportunity for cross-disciplinary applications of the research findings. Significance: the implications for space travel are particularly noteworthy as long-duration missions pose substantial risks from radiation exposure. Developing a method to protect astronauts could enhance the feasibility of future explorations such as missions to Mars. This connection between oncology and space exploration underscores the far-reaching impact of this research. 4. Engineering and immunogenicity challenges. The report mentions ongoing efforts to engineer a human-compatible version of the Dsup protein to minimize immune responses. This aspect may be glossed over, but it is critical for the transition from animal models to human clinical trials. The original tardigrade protein may trigger an immune response in humans, complicating its use in therapy. Significance: addressing immunogenicity is a common hurdle in the development of biologics. By focusing on creating a version of Dsup that is less likely to provoke an immune response, the researchers are paving the way for safer and more effective clinical applications, thereby enhancing the likelihood of successful human trials. 5. Potential for enhanced chemotherapy tolerance. The study suggests that the Dsup protein could also be beneficial in mitigating DNA damage caused by chemotherapy. This is a crucial point, since the side effects of chemotherapy can be just as debilitating as those from radiation therapy. Significance: if Dsup can be leveraged to protect against chemotherapy-induced damage, it could lead to improved tolerability of standard cancer treatments, allowing patients to maintain their treatment regimens without significant interruptions due to adverse effects. This could ultimately lead to better survival rates and quality of life for cancer patients. 6. Novelty of mRNA delivery systems. The innovative use of polymer-lipid nanoparticles for mRNA delivery is a noteworthy technical achievement. The combination of these two delivery systems enhanced the efficiency of mRNA transfection, enabling a robust expression of the Dsup protein in target tissues. Significance: the advancement of delivery systems for mRNA therapies is a rapidly evolving field. The findings from this study could influence the development of other mRNA-based therapeutics beyond oncology, potentially impacting vaccine development and other areas of medicine that require efficient gene delivery methods. Conclusion: the research on the Dsup protein from tardigrades represents a significant advancement in the field of oncology, offering a novel approach to protect healthy tissues during radiation therapy. However, the implications of this research extend far beyond cancer treatment. The temporary nature of mRNA delivery, the localized protective effects, the potential applications in space exploration, and the engineering challenges associated with immunogenicity are all critical aspects that underscore the transformative potential of this research. By recognizing and addressing these elements, researchers and clinicians can better harness the protective mechanisms of nature to improve patient outcomes in oncology and potentially other fields. The research on the tardigrade-derived protein Dsup and its potential application in shielding cells from radiation effects during cancer treatment represents a significant breakthrough in oncology. Here are 4 key points for in-depth analysis and discussion. 1. Mechanism of action: Dsup protein's role in DNA protection. The Dsup protein from tardigrades exhibits a remarkable ability to bind to DNA and suppress radiation-induced damage. This protective mechanism is crucial, especially given that radiation therapy, while effective for treating various cancers, often leads to severe side effects due to collateral damage to healthy tissues. The ability of Dsup to significantly reduce DNA strand breaks, demonstrated by a 50% reduction in DNA damage in mouse models, highlights its potential to enhance the safety and tolerability of radiation therapy. This raises questions about the underlying molecular mechanisms that make Dsup effective and whether similar strategies could be utilized with other proteins or in different therapeutic contexts. 2. Innovative mRNA delivery system. The researchers developed a sophisticated method of delivering mRNA encoding for the Dsup protein using polymer-lipid nanoparticles. This delivery system is significant for several reasons. First, delivering mRNA rather than the protein itself allows for temporary expression of Dsup without permanently altering the patient's cellular DNA, thereby reducing potential long-term risks associated with gene therapy. This innovative approach also underscores the increasing importance of mRNA technology in medicine, especially following the success of mRNA vaccines during the COVID-19 pandemic. A discussion could focus on the challenges and advancements in mRNA delivery systems, their potential applications beyond oncology, and the implications for future therapeutic developments. 3. Clinical implications and broader applications. The potential clinical applications of Dsup extend beyond just improving radiation therapy for cancer patients. Given its efficacy in protecting against DNA damage, Dsup could also be utilized to mitigate side effects associated with chemotherapy, which similarly damages healthy tissue. Furthermore, the research opens avenues for protecting astronauts from cosmic radiation during space missions, highlighting the versatility of Dsup. This prompts a broader discussion about how biological innovations inspired by extremophiles like tardigrades can inform solutions to diverse problems across fields such as space exploration. Environmental science and medicine. 4. Challenges and future directions. While the findings are promising, several challenges remain before Dsup can be used clinically in humans. One major concern is the immunogenicity of the Dsup protein. The natural protein may provoke an immune response in humans, necessitating the development of a human-compatible version. Additionally, optimizing the delivery system for scalability and ensuring its efficacy in diverse patient populations are critical next steps. Future research will need to address these hurdles while also exploring the long-term effects of Dsup expression and monitoring for any potential adverse effects. The discussion could explore the balance between innovation and safety in biomedical research, the regulatory pathways for new therapies, and the ethical considerations of using biologically derived substances in human medicine. In summary, the research on Dsup represents a promising intersection of biology and medicine, with the potential to transform cancer treatment and offer protective strategies for various applications. Each of the key points discussed provides avenues for further exploration and highlights the importance of collaborative research efforts in addressing complex health challenges. 1. The discovery of penicillin, 1928. The discovery of penicillin by Alexander Fleming marked a pivotal moment in medical history. Fleming noticed that a mold called Penicillium notatum had contaminated one of his petri dishes and was killing surrounding bacteria. This serendipitous observation led to the development of penicillin, the world's first true antibiotic, which transformed the treatment of bacterial infections and paved the way for modern antibiotics. The widespread use of penicillin during World War II significantly reduced infection-related deaths among soldiers and civilians, establishing antibiotics as a cornerstone of modern medicine. 2. The development of the polio vaccine, 1955. The development of the polio vaccine by Jonas Salk represented a monumental achievement in public health. Polio was a devastating disease causing paralysis and death, particularly among children. After extensive research and trials, Salk introduced the inactivated poliovirus vaccine in 1955, leading to a drastic reduction in polio cases across the globe. The success of the polio vaccine not only saved countless lives but also inspired subsequent vaccine development efforts, showcasing the power of vaccination as a public health tool. 3. The Human Genome Project, 1990 to 2003. The Human Genome Project was an international research endeavor aimed at sequencing and mapping all the genes of the human species. Initiated in 1990 and completed in 2003, this ambitious project provided a comprehensive blueprint of human DNA, enhancing our understanding of genetics, disease mechanisms, and human biology. The insights gained from the Human Genome Project have revolutionized fields such as genomics and personalized medicine, leading to advancements in targeted therapies and genetic testing that are fundamental to modern oncology and other areas of medicine. The recent study on the use of the tardigrade-derived protein Dsup in protecting healthy cells from radiation damage during cancer treatment marks a pivotal advancement in oncology. Given its potential, the future outlook for this research and its applications can be predicted across several dimensions, including clinical practice, technological innovations, and broader applications in medicine and space exploration. Clinical implications. 1. Enhanced radiation therapy. As clinical trials progress, the application of Dsup mRNA therapy could revolutionize radiation therapy for cancer patients. If proven successful in human trials, this treatment could significantly mitigate the debilitating side effects associated with radiation therapy, such as oral mucositis and hematological complications. This would not only improve patients' quality of life but may also lead to higher treatment adherence and better overall survival rates. 2. Personalized cancer treatment. The ability to use a patient's own cells to produce the Dsup protein could enable personalized approaches to cancer treatment. This could allow for tailored radiation doses that are safer and more effective, as clinicians could adjust treatments based on individual responses to the protective effects of Dsup. 3. Combination therapies. The Dsup protein's protective qualities could be synergistic with other cancer treatments, including chemotherapy. Research into combining Dsup with various chemotherapeutic agents could lead to innovative treatment regimens that reduce DNA damage to healthy cells, thereby enhancing the efficacy of cancer therapies while minimizing side effects. Technological innovations. 1. Nanoparticle delivery systems. The successful delivery of Dsup mRNA via polymer-lipid nanoparticles highlights the potential for further innovations in mRNA delivery technologies. Future research may lead to the development of more sophisticated nanocarriers that enhance the stability, localization, and efficiency of mRNA delivery, not just for cancer therapies but also for other diseases. 2. Immunogenicity management. Addressing the immunogenicity of the Dsup protein is crucial for its clinical application. Future research will likely focus on engineering human-compatible versions of the Dsup protein that minimize immune responses. This could open doors to broader applications of mRNA therapies by ensuring their safety and efficacy in diverse patient populations. 3. Advancements in mRNA technology. The Dsup study exemplifies the potential of mRNA technologies beyond vaccines, paving the way for a new era of mRNA therapeutics. Future developments might explore other protective proteins derived from extremophiles or other natural sources, which could expand the therapeutic arsenal available to clinicians. Broader applications. 1. Space exploration. The implications of Dsup extend beyond oncology. The ability of this protein to protect against radiation could prove invaluable for astronauts exposed to cosmic radiation during long-duration space missions. Future research may focus on how Dsup-based therapies could be integrated into astronaut health protocols, ensuring their safety during missions to Mars and beyond. 2. Environmental resilience. Research into the protective mechanisms of tardigrades could inspire biotechnological innovations aimed at enhancing resilience in other organisms. This could lead to advancements in agriculture, where crops might be genetically modified to withstand extreme environmental conditions, including radiation exposure. 3. Regenerative medicine. The understanding of DNA damage mechanisms and the protective roles of proteins like Dsup could inform regenerative medicine strategies. By minimizing DNA damage in stem cells or transplant tissues, Dsup may enhance the viability and functionality of transplanted organs or tissues. Conclusion. The pioneering research on the Dsup protein from tardigrades not only represents a significant advancement in cancer treatment but also opens avenues for diverse applications across medicine and technology. As research progresses, we anticipate transformative changes in how we approach radiation therapy, cancer treatment, and even space exploration. The integration of biological insights with cutting-edge technology will likely lead to solutions that address some of the most pressing health challenges of our time, ultimately improving outcomes for patients and expanding the frontiers of human capability.